Hey everyone. So we just finished talking about how cells communicated via messages. So whether that messaging was long distance, medium distance, or short distance. Now we're gonna start talking about the chemical, what's happening chemically behind this, and how this is gonna relate to the actual interpretation of that message that was being sent from one cell to the target cell. And so we're just going to do a brief overview. We'll go over the main points later. We're just going to cover the main points right now, and then we'll go into the details a little bit later. And so there are three main steps that are happening in cell signaling and how a cell responds to a message. And so if you can get these 3 points down, you are solid for this unit. So the first step is going to be reception. This is when a ligand is going to bind to a receptor. A key gets into the lock. Please get familiar with this vocabulary as it will be used pretty frequently in this unit. So like a key, the ligand will act as a key, and the receptor will act as a lock. The reception is going to be followed by transduction, and that is when the signal is going to be converted from one form to another that the cell can actually interpret. And so think of like stoichiometry. We are converting units to units to units to get a certain variable. Think of it like this. This is the cell's way of doing stoichiometry, or translating a message from like English to Spanish or Spanish to English. And then lastly, we'll have a response. And so the cell is actually going to take that signal and actually do something with it. And this could be a variety of things. This is just the brief, and now we're actually going to go into the detail. So we're going to start off with stage 1, reception. Key vocab, reception and receptor and ligand. Reception, the detection and receiving of a ligand by a receptor in the target cell. And so we're going to have a target cell. It's going to have a receptor, target cell, a receptor, and it's going to have a ligand. The ligand is the key that is not a part of the cell that's going to connect to it. It's going to be a perfect fit, and it's going to cause the reaction to happen. The receptor is a macromolecule that binds to a signal molecule, the ligand. All receptors have an area that interacts with the ligand and an area that transmits signals to another protein. What's going to happen here is that there's going to be a binding between a ligand and a receptor is highly specific. So like I said, like a key and a lock. Key to a lock. Let's move that somewhere else. Oops. Key to a lock. Very specific binding that's happening right here. And what we're saying, let's add on to this diagram that we have going on right here is saying that this receptor is going to be bound to some sort of protein. And that protein is going to be in charge of interpreting that message. So we have a target cell with a receptor. A ligand is going to come in, and this ligand is going to lock into the receptor, and that's where that signal is going to be transmitted, and it's going to be transmitted to the protein. Let's do color pencil, blue. Poop, 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 poop. The blue is going to be that signal. When that ligand binds to the receptor, that receptor is going to activate, and it's going to cause a conformational change. Which means that it's just going to change shape. This allows the receptor to interact with other cellular molecules and initiate a transduction signal. We're going to find most receptors to be on the plasma membrane, but some of them will also be intracellular, which means they'll be on the inside of the cell. So with plasma membrane receptors, these are the most common types. These receptors are going to bind to ligands that are either polar, water soluble, or large, things that cannot pass through the cell membrane, that have a hard time going through membrane. And some examples are going to include GPCRs, G protein coupled receptors, which we'll talk about in just a moment, and ligand gated ion channels. Think of the sodium potassium pump. And then we're going to have intracellular receptors that are to be found inside the cytoplasm or in the nucleus of a target cell. These bind to ligands that can pass through the membrane, so like hydrophobic molecules like hormones, steroids, and gases, and anything that's nonpolar, things that can easily pass through the cell membrane. When you actually take the AP exam, you do not need to figure out if a molecule is hydrophobic. We will tell you if it's hydrophobic, or they will say that this is a steroid hormone. And so if we tell you it's a hormone, you should be able to know that we are talking about an intracellular receptor versus a plasma membrane receptor. Next, we're going to have transduction. This is going to be our second step. And so we have the ligand that binds to the receptor, and it's going to cause a signaling to be converted. Transduction, keyword. Transduction is going to be the conversion of an extracellular signal to an intracellular signal that will bring about a cellular response. So basically like translating a language. And this is going to require a series of changes in a series of molecules known as a signal transduction pathway. This signal transduction pathway is going to regulate the protein activity through a couple different things. And so when we mean signal transduction pathway, in the conversion of this message, there's going to be a bunch of proteins that are going to be involved. And so we're going to have a couple things happening. We're going to have phosphorylation by an enzyme. When we phosphorylate something, we are adding a phosphate to it. So think of when we were talking about sodium potassium pump. To turn on the potassium pump, we had to phosphorylate it to turn it on, and when we need to turn it off, we need to dephosphorylate. We need to remove a phosphate, and that's going to shut off a pathway, whereas a phosphorylation is going to turn on a pathway. And remember, when we change the shape of a enzyme or a pathway or a protein, that's going to change the function of it, whether it's on or off. During this transduction, the signal is going to be amplified. Think of like a game of telephone. You tell 3 people, those 3 people tell 3 people, those 3 people tell 3 more people, and so that message gets sent across pretty quickly. And what's involved of amplifying that message is going to be second messengers. These are going to be small non-protein molecules and ions that are going to help relay and amplify the response. A common example is going to be cyclic AMP, or we call it CAMP. And so we said second messengers. Who's the first? Well, the first messenger is this dude right here. First messenger. He brings the initial message, and then the second messengers are going to be the ones that are going to go and tell basically everyone inside the cell. Not a good example. Let's try this. There we go. Finally, we're going to have a response. A response is the final molecule in the signaling pathway that converts the signal into the response that's going to cause a cellular process. And so, for example, this might be a protein that can alter membrane permeability. This could be an enzyme that will change a metabolic process or a protein that turns a gene on or off. So it's that final conversion. And so we're going to talk about how signaling cascade works, how like these messages get across. So we're going to have, let's look at this far image right here, number 1. We're going to have a pathway that is completely off. There's no ligand, so therefore the receptor and the protein are off. There's nothing happening. We're going to get a ligand. He is going to attach to the receptor, and he's going to turn on our protein, our initial protein, or he's going to activate the receptor. That receptor is then going to activate the protein that is involved in the signaling transduction. That protein is going to activate a protein in the cytosol, and that next protein is going to activate the final target. And this final target could be CAMP, which is eventually going to cause a final response, the response that we are needing. 4, 5, 6. Basically, the mess is like a game of telephone. One person tells another, another tells another, another tells another, and this message is going to be spread across the cell until it eventually causes that response. Hence, a cascade. And basically, how are we turning these things on? Turning on via phosphorylation. How can we change the signal transduction pathway? Well, one way to change it is through mutations. Mutations in receptor proteins or any component of the signaling pathway can cause a change in the transduction of the signal. So if we change something in steps 1, in reception, 2, transduction, or 3, response, that can change how the actual cell is going to respond to the message. It can happen at any step. It can also happen at any step here as well. 1, 2, 3, 4, 5, 6. So one example of a signal transduction pathway is going to be of GPCRs. And so let's first talk about the players. We're going to have G protein coupled receptors. This guy right here. So let's number these. And we got 1. We're going to have the ligand, which is going to act as the key. We're going to have our G protein, which is going to trigger the enzyme. We'll have our energy, GDP or GTP. Think of ATP, but it's another form for, it's basically just another form of energy. We're going to have the enzyme, which will trigger the secondary response, the secondary messengers. And then we'll have the secondary messengers, which will trigger the response. And so it's basically one thing turns on another. This turns on this turns on that, and etc. When we get GDP to GTP, I forget what the G stands for, but G diphosphate to G triphosphate, we are phosphorylating this GDP molecule. Phosphorylation, adding a phosphate. This might be easier to watch along with a video, just watching it step by step, but just know this causes this causes that, and it kind of acts like an avalanche. So let's go through the actual steps of signal transduction. with with, with the GPCR. So we're gonna have the first step, reception. The G protein is inactive. This is not active until it finds a ligand that is gonna connect to its receptor. This ligand is specific to this GPCR. Once the ligand binds to the receptor, it's going to be activated and it's going to change shape. It changing shape is going to cause GDP to turn into GTP via phosphorylation. When this GDP molecule is, before I get into that, save that for later. So review again, we're going to have an inactive GPCR. It doesn't have a ligand. Once that ligand actually binds to the GPCR, it's going to change shape and activate. This will cause the GDP to turn into GTP via phosphorylation, and this G protein is now going to be activated. The G protein is now activated and is now going to move towards the enzyme. And when that protein binds with the enzyme, it's going to cause the enzyme to trigger another reaction, and it's going to cause the secondary messengers to actually become active. It's going to activate them. And what's happening here is that we're amplifying the reaction. How one reaction can trigger another reaction. And so this is activating several secondary messengers. And then lastly, the response. The secondary messengers are going to influence a cell and elicit an intended response, so response that we needed. And this is the most common way that cells go through cell communication and how they interpret these messages that are received from other cells. Let's talk about ion channels. We already know about what the sodium-potassium pump does, but let's talk about it in the context of cell communication. And so we're going to call these ligand-gated ion channels, and these are going to be located in the plasma membrane, and they're important for the nervous system. The receptors are going to act as a gate for ions. When ligands bind to the receptor, the gate is opening or closing, which allows for the diffusion of specific ions, like sodium or potassium. And that will initiate a series of events that eventually lead to a cellular response. And so here we have the sodium-potassium pump, or an ion channel. To be more specific, my apologies, an ion channel. This will be turned off until it receives ACH, a neurotransmitter that will act as the ligand. This will bind to the receptor, causing this to change shape, and now will allow for molecules or ions of sodium and calcium and potassium to go up and down through the channel. Next, in the next video, we'll be talking about how our body is able to regulate internal conditions like temperature, blood sugar levels, blood pressure levels, etc., and how all this is going to connect to how our body maintains homeostasis. This might be a confusing topic right now with communication. I do recommend watching some videos that go through the process so that you can visually see what's actually happening. But ultimately, the three main steps that you need to know are going to be transduction, and response. If you are able to break down these three steps, you're good for this whole unit, in all honesty. So I will see you again in the next video.